Welcome back to the University of Melbourne Biology series. This is episode 9, and today we tackle one of biology's great puzzles: why do offspring resemble their parents, yet never copy them exactly? A pea plant crossed with another pea plant can give yellow seeds and green seeds in numbers so regular you can predict them in advance. How? The answer turns out to be hidden inside a special kind of cell division. Over the next 12 minutes we will start with that division, meiosis, then follow the trail it lays down, how single alleles separate, how we diagram a cross and read it with a Punnett square, how two traits behave at once, and finally how we trace inheritance through human families and put a number on it. Heredity, it turns out, runs on rules. Let us learn them. Everything starts with meiosis, a specialized cell division that reduces the chromosome number by half, producing haploid cells. Here is its signature feature. Meiosis runs as two rounds of division, called meiosis I and meiosis II, and the order of events is what matters. During meiosis I, homologous chromosomes, the matching pairs you inherited one from each parent, pair up and then segregate, with one member of each pair going to each new cell. Then in meiosis II, the sister chromatids separate, much as they would in an ordinary cell division. The end result of the whole process is a set of haploid cells, each carrying a single copy of each chromosome rather than the original pair. This having is not a flaw to be corrected, it is the whole point, the reason sexual reproduction can recombine two parents without doubling the chromosome count every generation. Keep your eye on meiosis I, because that step, where homologous chromosomes pair and pull apart, is about to explain the deepest rule in all of genetics. Now connect that division to genes. A gene can exist in different forms called alleles, and allelic segregation is the separation of those different forms of a gene during meiosis. This is the basis of Mendel's first law of inheritance. Think about what meiosis I actually did: it separated the homologous chromosomes. Because the two alleles of a gene sit on those homologous chromosomes, separating the chromosomes automatically separates the alleles. The consequence is simple and powerful. Each offspring inherits one allele from each parent, never two from the same parent for a given gene. So the choreography of chromosomes in meiosis is not just cell biology trivia. It is the physical machinery that makes inheritance predictable, and it is exactly what Mendel deduced long before anyone could see a chromosome move. Let us put segregation to work in a monohybrid cross, a cross that follows a single gene. To predict the outcome we use a tool called the Punnett square, which predicts the outcomes of genetic crosses based on allelic segregation. The idea is to lay out the alleles each parent can pass on, then combine them in every possible way. Because segregation guarantees that each parent contributes one allele per gene, the square systematically pairs every allele from one parent with every allele from the other, leaving no combination forgotten. Reading off the boxes gives you the offspring genotypes and how often each is expected to appear. The beauty of it is that the same simple grid works for any single gene, whatever the trait. The Punnett square is really just bookkeeping for segregation, a way of turning Mendel's first law into a concrete prediction you can check against real offspring. Predicting offspring is one thing, working backwards to figure out a parent's hidden genotype is another, and for that biologists keep a small toolkit of crosses, each answering a different question. A test cross identifies the genotype of an unknown parent by crossing it with a homozygous recessive individual. A back cross is performed to determine if a recessive trait is present in a population. And a reciprocal cross swaps which parent carries which trait, and it shows that the segregation of alleles is independent of their original pairing. Alongside these crosses, one more distinction matters in practice, telling apart traits carried on the ordinary autosomes from those carried on the X chromosome, because the two are inherited in different patterns. Different crosses, different jobs, all built on the same underlying segregation. The test cross deserves a closer look, because it solves a genuinely tricky problem. Imagine an individual showing a dominant phenotype. You cannot tell from its appearance alone whether it carries two dominant alleles or one dominant and one recessive, because in both cases the dominant trait is the one you see. The test cross cuts through that uncertainty by determining the genotype of that individual through a cross with a known recessive individual. Since the recessive partner can only contribute recessive alleles, it adds nothing of its own to hide behind, so the genotypes of the offspring reveal the allelic arrangement of the gene being studied. If any offspring show the recessive phenotype, the unknown parent must have been carrying a hidden recessive allele all along. The test cross is especially useful when analyzing dihybrid crosses, where we are tracking two genes at once, and that is exactly where we are heading next. Now we raise the stakes to two genes. A dihybrid cross involves two traits controlled by different genes located on different chromosomes. Here Mendel's second idea takes over, the law of independent assortment, which dictates that these two traits segregate independently of each other during meiosis. In other words, which allele a gamete receives for the first gene tells you nothing about which allele it receives for the second. The traits are dealt out separately, like two independent coin flips. This independence is precisely why a dihybrid cross produces predictable ratios of genotypes and phenotypes in the offspring, instead of a confusing jumble. There is one important caveat to flag: genes that sit close together on the same chromosome are linked and may not assort independently. But for genes on different chromosomes, independence rules. So how do we actually calculate those ratios? The law of independent assortment lets us predict the genotypic and phenotypic ratios in the offspring of a dihybrid cross. Because the two genes are independent, you can treat each gene on its own and then combine them. Picture the dihybrid cross laid out as a larger Punnett square, every gamete combination from one parent paired with every combination from the other. The independence of the two genes is what makes the offspring fall into clean, repeatable proportions rather than chaos. This is the real payoff of Mendel's two laws working together. Segregation tells you what each single gene does, independent assortment tells you how two genes combine, and from those two principles alone you can forecast the makeup of an entire generation before it is even born. Crosses are easy to set up in pea plants, but you cannot breed people on demand. To study human inheritance we instead use pedigrees, graphical representations of family histories that help us understand how genetic traits are passed down. A pedigree is drawn with specific conventions and symbols that represent each individual, their sex, and whether or not they carry the trait, then connect them across generations. The point of all this careful drawing is interpretation. By reading the pattern of who is affected and who is not, we can reveal whether a trait is inherited as dominant or recessive, and we can judge the likelihood of that trait appearing in future generations. This makes pedigrees a valuable tool for genetic counseling and for understanding the genetics of a whole population. Reading a pedigree naturally leads to a sharper question: what is the actual chance that a given individual inherits a trait? This is the probability of inheritance, and it is calculated from the genetic makeup of the individuals involved together with their family history. The probability is determined by the combination of alleles coming from both parents, exactly the combinations that segregation and the Punnett square laid out for us earlier. These calculations can be complex, and they genuinely require a working knowledge of Mendelian genetics and of how to read a pedigree. But the reward is enormous. Understanding the probability of inheritance is crucial for genetic counseling and for helping people make informed reproductive decisions. We have moved from chromosomes pulling apart in a cell, all the way to a number that can guide a family's choices. Step back and look at the single thread running through everything today. It began with meiosis separating homologous chromosomes. That separation is allelic segregation, Mendel's first law, which guarantees each offspring gets one allele from each parent. The Punnett square turned that law into predictions for a single gene. Independent assortment extended the logic to two genes at once in the dihybrid cross. The test cross let us read hidden genotypes, pedigrees let us trace the same rules through human families, and probability let us put a number on the outcome. One mechanism, scaled up from a dividing cell to a whole family tree. That is the quiet power of genetics, a few rules, applied faithfully, that let us predict the shape of the next generation. See you next time.